Okay, I think we're ready to start. I'd like to welcome everyone to the Kansi Stage International Speaker Seminar Series. This is our first one for 2025. Thank you for joining us. There's a couple ways that you can participate. There's questions can be put into the chat. And as well, if you have a very urgent question, you can perhaps put your hand up. But the speaker is willing to take questions as we go along. My name is Shelley Bull. I'm one of the co-directors of the Kansi Stage training program. One of our major activities or major things that contribute to the training program is this series of international speaker seminars. So the seminars themselves really aim to expose trainees to high caliber international research with the aim of further interdisciplinary research, innovation, discovery, and learning. We hope to foster a research community of genetic epidemiologists and statistical geneticists, particularly in Canada, to promote relevant and rigorous research in this field and to enable new networks of collaborative research. STAGE operates with the support of Kansi Ontario, which is an extra departmental unit in the Faculty of Arts and Science at the University of Toronto. And the seminar series themselves are sponsored by the Hospital for Sick Children, the Lunenfeld Tannenbaum Research Institute, the McLaughlin Center, the Princess Margaret Cancer Center, and the University of Toronto Department of Statistical Sciences. So I'm very happy today to introduce our speaker, Professor Josée Dupuis. She's currently chair of the Department of Epidemiology and Biostatistics and Occupational Health at McGill. She's been there since 2022. Professor Dupuis actually began her academic career as an undergraduate in statistics at Concordia University in Montreal, and then went on to graduate training in statistics at Stanford. Prior to return to Canada, Professor Dupuis was professor and chair of Department of Biostatistics at Boston University in the School of Public Health there. She has received numerous awards and honors, including leadership awards from the American Society for Human Genetics and the International Genetic Epi Society, as well as awards for mentoring and teaching. Professor Dupuis has been involved in various consortia and cohort studies, including CHARGE, TopMed, and the very well-known Framingham Heart Study. Among other contributions to research, she has maintained interest in methodology for family studies, and we're looking forward to hearing more about that today. So thank you very much, and we can go ahead now with the talk.
Thank you very much, Shelley, for this kind introduction, and thank you for the invitation. I'm delighted to be presenting some of my research on incorporating family history in rare variants association analysis. As was mentioned, feel free to raise your hand or if you have questions, so put them in the chat and they'll be monitored during the presentation. Okay, this work was supported by grants from the NIH, and I have no conflicts of interest to report. I'll start with a background on genome-wide association studies and rare variant analysis. Then I'll introduce or propose approach to incorporate family history in rare variant analysis that we call FAT, Family History Aggregation-Based Test. And then a couple of applications using UK Biobank data for all-cause dementia and hypertension. Genome-wide association studies have led to the identification of thousands of associated genetic variants, but most of the identified variants are common, not rare, and they have relatively small effect sizes. And the power depends on the number of cases included in the analyses and the ideal frequency of the variants. Rare variants, on the other hand, are crucial to understanding the unexplained heritability. And their association, because it's so difficult to detect association with rare variants, their associations are evaluated in groups of rare variants, not one rare variant at a time. And then finally, what I call history is the information that's collected on the status of relative. When you enter a study or when you go to the doctor's office, oftentimes you're asking, like, did your parents have heart disease? Did they have dementia? Did they have cancer? So that information is routinely collected by physicians and in other studies. And it can be incorporated to enhance statistical evidence. So genome-wide, I'm sure many of you are familiar with genome-wide association studies where thousands to millions of single nucleotide variants are genotyped on study participant, either case control or cohort studies. And what I call a SNP, the single nucleotide variant, is a genomic location where the genetic material may differ across individuals. When I give this talk to statisticians, I tell them to think of a SNP as a 3-category variable where the 3 categories are 0, 1, 2, and representing the number of minor alleles. Each SNP is tested for association. So even though we have millions of these, we analyze them one at a time for association with the trait of interest. And when we find an associated variant, it's not so much that it's causal, but it's assumed to be close to a gene that influenced the disease risk. So here's a representation. The SNP I often also called SNP for single nucleotide polymorphisms. And most of our genome is common to all individuals, but there are some locations where there are differences, and those are the part of the genomes that we're interested in. The challenge with GWAS is the multiple testing burden. So stringent thresholds, example, a typical threshold of 5 × 10⁻⁸ for the p-value is required to declare an association. And replication, I'm saying, is often sought, but is actually needed to validate findings. The other challenge is most genetic variants have a small effect on disease risk, and most studies alone are underpowered to detect such effect. And huge sample sizes, such as the ones in the UK Biobank with 500,000 participants, are required to detect association. And the other challenge is very low power to detect association with rare variants. And often, as I mentioned before, these are tested jointly to improve power. So here's a generic model for a genome-wide association study. As I mentioned, each variant is analyzed for association using mostly an additive model. In this particular model here on the left side, I have the phenotype Y, it's a vector of disease phenotype or quantitative trait. So that G function represents either linear or logistic regression to estimate the effect of the genotype. Oftentimes, we're adjusting for some known risk factors. So, for example, age could be one of the variable. Smoking status could be another variable if we're looking at diseases that are pulmonary in nature. But we're not so much interested in the effect of these variants. We just want to account for them. And what we're interested in is this G here, which is the genotype of each individual. Again, like a 3-category variable. If you're using imputed, it could be the expected number of minor alleles. And then we want to estimate beta, which is the effect size, the estimate of effect of beta on the disease or on the quantitative trait. And the p-values for this test, each SNP is analyzed one at a time, is usually presented as a Manhattan plot. I'll give you an example for type 2 diabetes. More than 400 loci have been associated with type 2 diabetes, but again, these 400 loci only represent a small portion of the disease heritability explained. And most of these associations were found after a meta-analysis of large number of studies, either with the diagram consortium, which is diabetes genetic replication and meta-analysis, or the MAGIC consortium, which is meta-analysis of glucose and insulin-related traits. And because of patient confidentiality, you can anonymize data, but once you have the whole genome of someone, it's no longer anonymous. So because of patient confidentiality, meta-analysis is a more feasible option than pooling data. So oftentimes each study will do their own analysis, and then a meta-analysis will be done, aggregating the finding at a SNP level. And the results look something like this. This is the result for fasting glucose, looking at the entire genome. So on the y-axis, you have the minus log 10 of the p-value. The higher, the better. Notice that this axis goes all the way to 70. So a p-value of 10 to the minus 70 is pretty impressive. This is a meta-analysis of 21 studies with over 46,000 participants. The little blue-purple variants were found in a single study. So those were a strong enough association. And then the black were found in the 21 studies, and actually the red were only found when we increase to even more studies, even more participants. So very large sample size, and each of these variants explained less than 1% of the variance in fasting glucose. So rare variant contribution has been really important for complex traits. Think of cystic fibrosis, or to Mendelian trait, think of cystic fibrosis and other monogenic disease. And these variants were identified through linkage and typically were in gene exons. But the contribution of rare variants to complex disease is not as well understood. Common variants identified so far only explain a small fraction of the total heritability. Rare variants are often excluded from GWAS because of low power. In the plot that I've shown you here, variants with frequency less than 1% were not analyzed. And so the unexplained heritability, I say that a lot of the a very small proportion of the variance in the trait is explained by the common variants. So the unexplained heritability may be caused by may be explained by the multiple rare variants. There are some pretty standard approaches for analyzing rare variants. And because of low power, they're often analyzed jointly. The most common approaches, and there are many, many out there of these approaches. I'm going to concentrate on a few. The most commonly used approach are a burden test. So instead of looking at the association of a single variant with the trait, you aggregate the sum of the rare allele that a person carries. There's also the SCAT test, sequence kernel association test, which is an aggregate score test for each variant in a variance component. And then there's a SCAT-O, which is kind of a optimized linear combination of the burden and the SCAT. And you have, these are not my methods. I'm just presenting the work of others to put my work into context. The model for the rare variants is exactly the same as it was before. This is the exact same model. The only difference here is that G is a matrix of genotypes. So instead of looking at a single genetic variant, we're looking at multiple genetic variant at a time. And instead of a single effect size, we're estimating multiple. So if we have K variants in that model, we would be estimated K effect size or K beta coefficient. And the hypothesis of interest is that all of the betas are equal to 0. And the specific form of the null hypothesis depends on the approach used. So pretty much every single rare variant analysis can be put into this general framework. For the SCAT, it's a joint effect of multiple variants in a region on a phenotype is tested, and it's why it's so popular is that you can include covariates, and it has a closed form, not a closed form, but an easily estimable distribution so that you can get p-values without permutation. So the general model is the same. What differs with the SCAT is that the assumption that the effect size follows some distribution F with a zero mean and a variance that's proportional to WK times tau. So the distribution does not have to be pre-specified. So I'm not assuming that the betas are normally distributed or anything like that. I'm just assuming they have some distribution with mean zero and a variance component tau. And I'm allowing that variance to differ by each variant by the incorporation of a weight for each variant. The weight is this WK. Note that the weights are not estimated in SCAT. Usually people will pre-specify the weight. And the weight is usually, when people apply SCAT, usually the weight is a function of the minor allele frequency, believing that the more rare variants are more likely to be deleterious. So the more rare a variant, the more weight you would give it. And the test is that all of the betas are equal to zero, and this is equivalent to simply testing that the variance is zero. Because if the variance is zero, that means that all the beta k's are equal to zero. So these two hypotheses are equivalent, and that's the test that was proposed by Michael Wu et al. To test a group of rare variants. So the variance component statistic as this form on the left, which is the, and it's really intuitive. So y minus mu hat is the residuals from a regressing y when you have a quantitative trait on the covariate. So it's the residuals from regressing y on the risk factor that you know. G is your matrix of genotypes. W is the weight matrix that's pre-specified. And you can actually rewrite this ugly matrix expression into simply, it reduces to the sum of the score statistics for each variant when they're in a model by themselves times their weight. So the score statistic is just the residual times the genotype. And this SCAT statistics is just a weighted sum of the individual test of each variant, which is really great because that means that if I only have summary statistics from these variables, I can still compute this SCAT statistics to test the association of multiple variants with the phenotype. And the other thing that's beauty here, it also, because you have these K component for the K SNPs, you can see which of the variant contributes the most to your test statistics. And under the null hypothesis that all of these betas are equal to zero, then the Q follows a distribution which is a weighted sum of chi-square distribution and can be estimated without the need of permutation. It's very efficient. Okay. It turns out that the burden test is also in this very general model. So again, my general model, the burden test assumption is that the beta case, each beta, is equal to beta times a certain weight. So the model reduces to having the phenotype on the left, the known risk factors, plus you have this weighted sum. You're creating this core, which is the weighted sum of the genotype, and you're estimating a single beta value, okay? And it turns out that the burden can be written, the burden test can be written also in terms of the single variance score statistics, but this time instead of having, before I had WK square SK square, like here, WK square SK square, this time is WK SK squared, okay? So what this means is that this is going to be most powerful when all of the variants have the same direction of effect, okay? So, but I don't know. So burden is most powerful when the genetic variant are all in the same direction, assuming the rare variants are all deleterious, they all increase your risk, or they all make you healthier and they all decrease your risk. But the direction of effect is usually unknown. I've yet to know a disease where I know the direction of effect. So um, to deal with that, because SCAT deals will do well, because SCAT, you're squaring the statistics, so the direction of effect is not affecting the variable as well. So the SCAT will do well when you have both positive and negative direction of effect, but the burden will be most powerful when all the effects are in the same direction. The SCAT-O, this optimized combination of SCAT and the burden statistics was proposed, where it's just a mixture of a combination 1 minus rho plus rho times the burden, and rho is between 0 and 1. And the p-value for each Q is computed, and the minimum p-value is taken as the SCAT statistics. And again, even though this is maximized over rho, it turns out that it's pretty easy to, there's a I'm saying closed form, but it's not accurate here. It's a computational form that's very efficient to get the p-value. Okay, I've told you everything that everybody else has done, but I've yet to tell you what we have done. So what we wanted to do is exploit family history in doing these joint tests. So the power of the GWAS depends on sample size and the number of cases. Some age-related diseases may have very low prevalence. And at the time I was working on Alzheimer's disease, which is a late onset disease. So when you look at things like the UK Biobank, the prevalence of Alzheimer's disease in the UK Biobank is very low. People who have Alzheimer's disease, by definition, may not participate in genetic study. However, the disease status of the parents may provide information on the future phenotypes of people who may not have yet developed the disease. And incorporation of family history may improve power especially when the number of cases is low. So let's assume we have family history of the disease status for relatives, but we don't have their genotype available. So I'm going to call Y capital superscript R. This is the phenotype of a relative of the participant in the study that I'm looking at. And I'm going to denote the phenotype, genotype, and covariance of the cohort participants as Y capital P, Y superscript P, G superscript P, and Z superscript P, respectively. I can write the likelihood of the phenotype of the participant and the phenotype of the relative in this way, conditional on the genotype of the participant, the risk factor of the participant, and the risk factor of the relative. I can break it down into two parts. The first part is or typical likelihood of the participant given its risk factor and genotype. And then the second is the phenotype of the relative, conditioning on the risk factor of the relative, the genotype of the proband, and the trait of the proband. So we assume that the phenotype of the participant is independent of the phenotype and covariance of the relatives after conditioning on the participant genotype. So it's a pretty big assumption. But what this means is that I have this likelihood here. I have two likelihoods that are independent. Notice here what's important is that I am conditioning on when I'm analyzing the relatives, I'm conditioning on the phenotype of the proband, and it's an important piece to make these two likelihoods independent. So here's the full likelihood, which has the trait of the proband and the trait of the relative. And the relative here, that could be mother, that could be father, that could be a sibling, that could be anyone. And I can model these into two probabilities, just the way I've done. Typically, this is done when you're asking for family history. Typically, it's saying did your mother have Alzheimer's disease? Did your father have cancer or something like that? It's typically done with the disease status 0 and 1. It's very rare you'll ask, like, what was the blood pressure of your mother? Those are not something that are collected. So even though the approach works with both quantitative and binary traits, here I'm focusing on the logistic regression. So I have my typical logistic regression for my proban, which is the trait of the proban on the genotype of the proban. And then I'm doing a second model, logistic model, looking at the phenotype of the relative. But this time I'm using the genotype of the proban. And I'm using the conditioning also or adjusting for the phenotype of the proban. And you can show that the relationship between the beta that I'm obtaining from this regression on proban phenotype and the beta that I'm getting from regression on the relative is there's this relationship between, there's an attenuation when looking at the phenotype of the regression. And of course, there's an attenuation. I don't observe the genotype of the relative. I only observe the relative of the proban. So it's kind of like measuring the genotype of the relative with some error. And that error is going to dampen my effect size estimate. So that the effect size estimate of the relative is approximately equal to 2 times phi, the coefficient of relationship, times the beta coefficient of the regression of the proban. So for first degree relatives, you would expect your beta first degree relative, the phi is equal to 1/4, 2 times phi is 1/2. So you would expect the beta of the relative to be 1/2 of the beta that you get of the proban. So if you listen to anything of my talk, if you remember anything of my talk, this is the most important one, is that what you get from, I'm sure you've seen in the literature, people call it proxy phenotype. They're using the proxy of the parent as the phenotype. And this is perfectly fine, perfectly legitimate, but I've seen meta-analysis where some cohort have a proxy phenotype, similar to having the YR here, and other cohorts have the real phenotype, and they're meta-analyzed together. It's a valid approach. Under the null, the approach is valid, but the effect size estimate is biased, and it's not the most efficient way of combining what you get from a proxy cohort, proxy phenotype, and others that you get from a real cohort. So if you remember anything of my talk is when you hear about proxy phenotype, that you know you need to do something to those estimates before you meta-analyze them. And these two estimates are independent under the assumption that I've talked about. So in the family history aggregation test that we're proposing, our FAT, we are getting 2 independent score statistics where we test the association of multiple variants with the participant, and we do similarly testing the association with the relative phenotype, and use meta-analysis to combine the scores from the phenotype of the participant and the relative, discounting the downweighting the score of relative by this 2 phi to account for the fact that the estimate is the relationship between the estimate that we get from the relative and the proban. And the FAT statistics for a binary outcome using logistic regression looks a little bit like this. It might be a little intimidating, but all it is, this is the residual from the regression of the proban, and then this is the genotype of the proban, the weight matrix, which is assumed to be known, residual from the relative phenotypes, genotype of the proban, not of the relative, times this W matrix, and the discounting factor. And this statistics also follows a weighted sum of chi-square distribution and is very quick and efficient to estimate. We can also rewrite, so this is the same statistics that I've rewritten. I've just taken the GPW outside. So here, GPW is on both of these terms. I can take it outside of this. So I just took this GPW outside, and this was transformed. So the W matrix transform is a diagonal matrix of weight. So the transform is the same. And I can rewrite it the way that uh, it was done for as a sum of the score statistics for the proband and the score statistics for the relative. So this QFAT can be written as weight square, the weight that I put on the variant, times the score of the proband, plus weight square times the score that I put on the relative, where these score statistics are the association of a single variant in the two regression model that I presented before. Okay, which allows me to have a fat burden analog to the scat burden analog. And so now instead of having the square of these things, I square the sum of these things, which is analog of running a test with a weighted sum of the genotypes. Now that I have a fat burden analog, I can have a fat O analog to the scat statistics, which is a linear combination of the family history aggregation test and the fat burden. And this can account for, and this is, again, has a very efficient way of estimating the p-value for this statistic. Note that this can you can account for relatedness among participants in the statistics. There's a scat version that takes an empirical or a kinship matrix that is known, so you can use that FAMSCAT when analyzing the getting the Q statistics and incorporate known or empirical kinship matrix. Can account for relatedness among relatives with known relationships. So unfortunately for the relatives, since you don't have genotypes, you can only account if the relationship is known. What motivated this work actually is work on the Framingham heart study, where I had phenotype information on all-cause dementia from the original cohort, lots of data, lots of cases, but I didn't have genotype. So I knew the relationship between these individuals, and I was able to correct for those. You can also adjust for unbalance in case control ratios. So in logistic regression, if you have many more cases than control, you might get inflated type 1 error. And that's also the case for SCAT if you have an unbalance of case and control. When you have a case control design, it's not so much a problem, but when you're looking at data like the UK Biobank and you're looking at things like dementia, the case control could be very imbalanced. And Zhu et al. Proposed a way to calibrate the distribution of this SCAT statistics using a saddle point approximation and efficient resampling called the SAGE method. And you can actually do the same thing with fat. You can do a saddle point approximation to calibrate the fat approach as well. But I'm not presenting the details here. Okay. So what are some of the alternative approaches to incorporate family history? Well, there's a group that proposed in 2020 a test for liability threshold model for family history, and I'll call this LTFH. What it does, it computes a posterior mean probability of, or mean genetic liability under a liability threshold model, conditioning on both case control status and family history. So I'll give a little example in the next slides. But what it does is that it gives you a genetic liability and it increases your liability if all of your relatives are affected, and it kind of decreases your liability or your risk of disease if all your relatives are not affected. And they've shown that this increased power in single variant analysis, and we've extended this LTFH method to work with rare variant analysis. So, and it's very, the only thing is that this LTFH is very computationally intensive, and it can only include, so far, the implementation first degree relatives. So how does it work? So this posterior mean liabilities, let's say that you have a control. So typically in a typical analysis, the control are coded 0, 1, 0, and your cases are coded as 1. So maybe you have a control who has no affected parents. So that's what I'm depicting on the top left. So maybe the fact that this person has no affected parent would decrease their risk of having the disease. So they may have a liability of, instead of being at 0, they may have a liability of minus 0.1. Let's say you have another control, but this time that control has an affected parent. So their risk increases, and instead of a phenotype of 0, maybe their risk increase and their liability would be 0.39. And then here's another case where you have a case which would be a 1, but both parents are affected. So their liability of disease might increase, let's say, to 1.56. So the 0-1 phenotype is kind of adjusted by the number of affected first-degree relative. And you compute this genetic liability conditional on the disease status of the first-degree relative. And then you can use that genetic liability, the minus 0.1, the 0.39, as a quantitative trait and a phenotype in your typical genetic analysis. So we went ahead and we compared these various methods for rare variant analysis. We looked at type 1 error and we looked at power. We compared the power of FAT and FAT-O, which takes family history, SCAT and SCAT-O, which do not take family history, the burden test. We included also the ACAT, which I haven't talked about much, but it's based on single variant association statistics, and it's a way of combining them, and it's very efficient. And then we implemented this liability threshold family history that I described on the previous page with SCAT and SCAT-O to do another test of burden of rare variant accounting for family history. And I'll present also an application to the UK Biobank. So for the type 1 error simulation, we simulated the trait of the participant, of the mother, and of the father, and it only depended on age and sex. It didn't depend on any genetic variant, although the trait was heritable. We had a heritability of 50%. And we converted this continuous trait, this continuous liability that had a heritability of 50% into a binary trait depending on which prevalence of the disease I wanted. So I have this trait that was simulated. If the simulated trait exceeded the cutoff, then this person was a case, and if it didn't exceed, that person was a control. And we had 5,000 participants with available parental history, did 10,000 simulation replicate, and did not implement the SAGE approach. So these were not corrected for imbalance in case control. And we looked at prevalence of 10, 20, 50% in participant, and the prevalence in the parent dependent on the heritability. And we looked at significance level of 5 times 10 to the minus 4 and 5 times 10 to the minus 6. And 5 times 10 to the minus 6 is like similar to what a correction you might do if you were analyzing every single gene on the genome using an aggregation method. So what we found, and what you're looking at is the empirical type 1 error divided by the significance level. So you want this to be 1. When it's greater than 1, then it's inflated. And as you can see, when you have imbalance, I have 10% of prevalence in proband. So that means for every proband, I have 9 controls. So great imbalance in the number of cases and control. All of the methods, no matter what the significance level looked at, all of the methods were inflated. And SCAT-O was the most inflated and the inflation is worse as you go down in the tail. So the more stringent the significance level you look, the more inflated it becomes. And SCAT-O was the one that was the most inflated. When you're at 50-50, all of the methods pretty much had the right type 1 error. So this inflation is due to this imbalance. Even when you were at 1 cases for 4 controls, you still had inflation. Not so much some of the tests at the 2.5 10 to the minus 4 significance level, but definitely in the tail, you still have issue. So we implemented the saddle point approximation, and we have a robust version for scat and fat. And when we did that, so again, you're looking at the type 1 error rate divided by the significance level. And here we have a very extreme prevalence of 5 in or 1 in for every 19 controls. And what you can see is that the robust scat has the right type 1 error at 0.05, 0.01, 0.005, the robust fat also. So all of the robust tests that use this saddle point approximation have the right significance level. And the non-robust version, they're doing not too bad at the 0.05. So when you don't have multiple testing, you're doing okay. But as soon as you go down to lower significance level, you see the inflation. Then we did some power analysis, again, looking just at participant, mother, father. And this time, like all of the variants together explained either 2% of the heritability and 2% when all the causal variants had positive effect, and then 3% when half had positive effect and half had negative effect. And again, age, sex had some were risk factors. And we converted the binary phenotype using a normal distribution. So 5,000 participant, 1,000 replicates this time, and we varied the number of variants in a region, the proportion that were causal, the direction of effect, if they were all positive or 50-50. And this time the disease prevalence is 20% for all of the simulations. I'm only presenting the 20% because of time constraint. So looking here, so here what we have is when most of them, everything has a positive effect. So that's what this plus is, how many had positive, how many had negative, and how many had no effect. So in the last is the best case scenario. All of the variants had a positive effect on the phenotype. And here only 80% had a positive effect and the other had no effect. So everything in the right direction, 40 variants. And what we see is that incorporating the family history either with the fat or the fat liability threshold family history really gain in power when adding the family history. And that the other tests like the burden, the SCAT-O, were much less powerful because of not incorporating family history. And what you can see also is when everything is in the same direction, the burden has a much higher power. This is the power of the SCAT, the yellow, and the burden is the gray. So when everything is in the same direction of effect, the SCAT is not as powerful, but SCAT-O really, that linear combination between burden and SCAT recovers this power. And this conclusion also holds true for when we had fewer variants, like this is 10% of the variants are causal and 90% are not causal. So the pattern is the same, just the power, the fewer variants, proportion of variants are causal, the smaller your power is. And this is the case where we had 50% positive and 50% negative. Again, really adding the family history is helping your power. But this time the fat or the FAT-O, whether you use fat and SCAT LTFH or whether you use FAT-O or LSCAT-O is the same. You're not getting much power by that optimization of versus SCAT and burden, and that's because your SCAT-O statistics is pretty much the same as the SCAT statistics. Your burden has very low power. And when all of them are causal, you see really a decrease in power, but incorporating the family history is really increased power. And the same pattern is true for the smaller proportion of variants. Here we have more variants that are non-causal, and the pattern is the same. I want to end with application. So I'm not selling fat. I'm telling you it has the same power as this LTFH um, uh, uh, analysis, but this is the time in minutes to analyze 1,000 region. So the fat takes about a minute to 2 minutes, and the LTFH method takes about 9 hours. So yes, they have both powers, but one is much more efficient than the other. So I want to end with an application to the UK Biobank. We looked at two phenotypes, all-cause dementia and hypertension. The mean participant age was 57, and the mean age of the mother and fathers was around 78. Information was collected on both parental history of all-cause dementia and hypertension. I took a trait with very low prevalence in the UK Biobank. So the prevalence of all-cause dementia in participant of the UK Biobank was 0.02%. However, this went up to 8% in mothers and 4% in fathers. And then I took a trait like hypertension that had about the same prevalence. And what we expect is we don't expect much gain in power by adding that information on hypertension, because the proband, which is going to be the biggest component in this statistics, already has a really decent sample size. But I expect a gain in power in the all-cause dementia because I have very, very few cases in the participant, but many more cases in mothers and fathers. What we took is we took genes that had common variant implicated. It doesn't mean that they had also rare variants, but we wanted to start somewhere. So these genes are implicated in all-cause dementia, and they have common variant. We looked at rare variants, and what we found, and when we compared SCAT or ACAT, is that we found much more evidence in using either the LTFH or the FAT method for rare variants also having involvement in these diseases for these four genes. When we looked at hypertension, we did not see that increase in power as we were expected. When we looked at incorporating the family history, SCAT was already detecting this gene as being associated with rare variants, and we didn't gain that much power by looking and incorporating the family history, which is what we expected. We also did a exome-wide association, and these are the top genes that came up. The blue ones are genes that have already been implicated with common variant in all-cause dementia. And similarly for hypertension, these are the top genes, and two of them have been implicated with hypertension. So in closing, inclusion of family history does improve power, especially for rare diseases. Our new approach and SCAT using the liability threshold lead to similar results. Both have increased power and more power than SCAT, but FAT is much more computationally efficient. The LTFH method can only incorporate first-degree relatives, and we've done simulation to show that incorporating more distant relatives will also improve power, but these results are not shown here. If you have case control imbalance, it's really important to use the robust FAT that has the correct type 1 error, and the robust SCAT as well implemented in the SAGE software, especially for low significance levels. And I want to thank my collaborator, Yan Bing, who was a student at Boston University, Han Chen, Gina Peloso, and Anita Stefano, our colleagues at Boston University. And I'll be happy to take questions.
Great, that was very well timed. So we have some time for questions. I think either people can put them into the chat or you can put up your hand. Mhm. I can start off with a question. So I guess one question with these kind of approaches. So I guess the most challenging case is the one where you just have phenotype on the family members. But I wonder, in some cases, you might have phenotype for some family members and phenotype plus genotype for other family members. So would that is that an extension that could be made?
No, actually, because if you can have some, there's no matching here. It's two separate regression. So if you have relatives that have both genotype and phenotype, you would just put them in the regression with the participants. And you
Uh,
would account for their relatedness that way. And then they just wouldn't have any relatives in the relative. So the everybody with genotype go in that participant regression and you account for relatedness using FAMSCAT or something like that. And then all of the participant where you only have phenotype go into the regression or the model with the participant, the relatives.
okay, thank you. Yeah. Andrew has a question. Do you want to just unmute and state your question, Andrew?
Yep, thanks, Josie. In the slide that you showed for UK Biobank, the dementia prevalence appears to be higher in mothers than fathers. Um, I guess my question is this consistent with known epidemiology, or could it be due to survival, sex differences in survival, or some aspect of reporting, family history reporting differences?
Great question. I do think that part of it is due to women are blessed with living longer than men still. So, meaning that more time to develop these all-cause dementia. But I don't know how much of this can be attributed to recall bias or that survival bias. You can see here that in terms of age, the age are very similar between the mother and the father, maybe slightly older in women. But it's a great question, and I don't have an answer. I would be more surprised, though, by the other way around. I would have been more surprised to see the prevalence higher in father than in mother, but I don't know how much this can be explained by survival bias or other recall bias or known epidemiology.
Thanks.
Okay, Michael Edwards, you have your hand up. Do you want to ask a
Yeah,
question?
I intended to ask another question, but following up on the last one, my first guess would be higher mortality from cardiac reasons for fathers, but that's just a shot in the dark on that one. Okay, I had a stats question. When you do the combined test, you're looking for uh, a, um, a p-value which is the minimum overall values of the weighting factor rho,
Mhm.
right? I wonder what is the thinking of why we looking at for the minimum p-value
Yeah.
instead of maximum p-value.
Well, you want the minimum p-value because you want the smaller, the smallest, like the evidence for association is with a small p-value. The smaller the p-value, the more association you
I would have
have.
thought, since you don't know the rho, that you would want to be conservative and say, well, what's the worst I can do? Use the maximum. I mean, that's my thinking anyway. When I look
Yeah,
at
no. Here is like what's the reason for doing this is that if all of your alleles are in the same direction, and believe me, I'm like this comes from the SCAT statistics. So if we go back, I'm sorry if I'm giving
Mhm.
everyone a headache, but if we go back to the SCAT O
Mhm.
statistics, which is here. Okay, so this is not my thing, but the thinking is when every variant is in the same, I have an effect in the same direction, scat is under power. Burden is doing much better. So in the case that every variant in the same direction, you would want your statistics, the Q row, to be rho equals 1, to be entirely burdened. But in the case where you have 50% positive and 15% negative, you want your statistics to reflect the Q scat because the Q burden is not good, doesn't have a high power. So you would want your rho to be 0 or your 1 minus rho to be 1 and your statistics to be equal to scat. So you can think of it this way. When you have everything in the right direction, the most powerful method will have Q equal to Q burden. And when things are 50-50, the most powerful statistics will have Q being equal to Q scat. And the way to achieve that is you could maximize the statistics. You can maximize based on rho, but it turns out that maximizing is not always equivalent to finding the most significant one because of the funky distribution of Q scat and Q rho. So that's why the authors of the scat O proposed to find the best, the optimal one that would minimize the p-value instead of maximizing the statistics over rho. I hope this gives you a little bit of intuition why that is.
That's right. Is it also discussed in the paper where the scat method comes from? So
yes, so this Lee paper that you have there would be where to go. We just mimicked what they've done with the fat.
Yeah, I'd like to read that and understand it better. Right, thank you.
Irfa, you have a question. Do you want to unmute and ask your question?
Yeah, I'm just curious, like I've seen a lot of these studies on like genetic related diseases, right? Like everyone does quite a few of them, but I'm just curious how how do they play in like population health sciences? Like, are they just merely theoretical in nature, or they actually drive some prevention, or they drive certain decision making? I mean, it'd be useful if it were if they were the latter, right?
Yeah, I think that like 23andMe, the genetic testing company, which I think is not doing so well financially or went bankrupt, wanted you to believe that you can make decisions on that. The effect of each of these variants are so small that they are not, I don't think they're yet used for prevention. That being said, I think the best example, there's an example of a drug that targets one of the genes that were found by GWAS. So what we can use, and I know what pharmaceutical company use the results of these identifying genes and all that to find drug targets to try to develop new therapy. So no, they're not used in the clinic in terms of prevention, but they're used in drug development.
Okay. So, Laurent, you have your hand up. Do you want to unmute, ask a question?
Yes, so it's a question that is a bit related to also Shelly's question is, yeah, if you could get more power by trying to impute the genotypes in the relatives. So for example, using segregation-based analysis or multiple imputations.
That's a very good suggestion. So especially if you have multiple related individuals, one could impute based on the related individuals and then just use these imputed. And I think intuitively you would do better, because right now what you're doing is you're just imputing the genotype of the participants without taking any, like one at a time, without taking any LD, anything into consideration. So I think this is a very intriguing suggestion, and especially if there are multiple relatives, you might be able to do a better imputation and gain better power in your the relative portion of your likelihood by using imputed genotype if you have multiple. Like if you have only one relative, you can't do better than just impute it to one of the relative for the relative. But if you had multiple relatives, I think you could improve on this idea.
And my second question is if you use some particular annotations to define the weights.
No, I have not. But I'm working with multiple groups who have, like, the latest is using a principal component of multiple annotations to incorporate. So I think there's a lot of room. Right now, all of my methods have used a simple, like the scat weight, which was like a beta distribution based on the minor allele frequency. All of these simulations are very, very simplistic.
Okay. Thank you.
Any more questions? Could maybe fit one more question in. Yeah, I'm just curious about whether there's information that could be extracted from the analysis about the actual betas. I mean, I understand that the approach is mainly about detecting genes, and that, it's usual that investigators would go on and, do some kind of, perhaps some fine mapping analysis, which I guess is going to be way more challenging for rare variants. But, there's an implicit sort of variant effect. Yeah.
Yeah. So from this little statistics in the scat and maybe in the fat as well, by writing the statistics in this way, you can then go look at what contributes, what's the biggest contribute. So this is a variant by variant contribution to the score. So you can go see which variant contributes the most to the score. So, so some people sometimes do remove one variant at a time and see what happens to the statistics. It's a lot of computation. You can look at this Q and you can plot this for each variant and see what's the highest, and it can point you to what's contributing the most, which rare variants or rare variants are contributing the most to the test statistics. And I think that's very informative. But there's no effect size estimate in a score approach method. You just have the contribution to the test statistic that you can use to try to identify which variants are the most important in your significant results.
Okay, thank you. Any last questions? Nope, well, that was a great discussion. Lots of very interesting questions and very helpful response.
Great ideas. Thank you.
So I'd like to thank our speaker for a really clear presentation of methods and really interesting applications and simulation studies. And thank you very much. And thank you for all the attendees and the great questions and your attention. And so we'll see you next time.